Hello everyone, welcome to our channel PHARMA GLP. In our previous video, we discussed the basics of stability studies as per ICHQ1A R2 guideline. Now in this video, we will go deeper into each aspect of ICHQ1A R2 guideline for stability testing of new drug substances and products. Also, we will find out the answers of our remaining questions like what are climatic zones, what is testing frequency and storage condition. So, stay tuned and watch this video till the end. And before moving ahead, please don't forget to hit that subscribe button and ring the notification bell so you will not miss any of our upcoming videos. Now let's start our today's topic. So friends, in continuation to our previous video, the stability can be defined as it is the capacity of drug substance or drug product to remain within specification limit and to maintain its quality such as potency and purity throughout its retest period or shelf life. When the term comes retest period, which means we are discussing about drug substances or API, and when the term comes shelf life, which means we are discussing about drug product or finished products. We have already discussed in our previous video about what is stability study, why stability study is required, what is the stability procedure in brief. If you have missed that video, please check it out before continuing with this video. I have given a link of that video in the description below, also in the I button. So, as we have discussed in our previous video, the stability study is required to understand product characteristics, to establish degradation pathways. This data is helpful for analytical method validation study, to establish expiry dates such as retest period or shelf life, to check compatibility of container closure and transport system, to recommend storage conditions, to design formulation and manufacturing process. It is a CGMP requirement, and in continuation to that, the most important objective or reason is the stability studies are also a regulatory requirement for product registration. And without having sufficient stability data of that particular product, we cannot even register or sell our product to the regulatory market. So friends, each organization should have established stability study procedure for product registration. Also, to ensure the safety of product throughout its shelf life if it is stored at a labeled storage condition. The stability studies will not only streamline the registration process but also help us to deliver safe and effective product to the market. So, the general principle of stability study is to provide evidence on how the quality of drug substance or drug product varies with respect to time, temperature, humidity and light, and to establish a retest period for drug substance or shelf life for drug product and to recommend storage conditions. So, before moving ahead, first let's understand what are climatic zones. The world can be divided mainly into 4 climatic zones, that is, Zone 1, 2, 3 and 4. These climatic zones are classified based on mean kinetic temperature. First climatic zone is temperate zone, second climatic zone is subtropical zone, third climatic zone is hot and dry zone. The fourth zone is further divided into 2 subparts such as Zone 41A and Zone 41B. Climatic zone 41A is hot and humid region and climatic zone 41B is hot and very humid region. Now these are the temperature and humidity conditions for these climatic zones. For zone 1, the temperature is 21 degrees centigrade and humidity is 45% RH. For zone 2, the temperature is 25 degrees centigrade and humidity is 60% RH. Then for zone 3, the temperature is 30 degrees centigrade and humidity is 65% RH. For zone 41A, the temperature is 30 degrees centigrade and humidity is 65% RH. And for zone 41B, the temperature is 30 degrees centigrade and humidity is 75% RH. RH means relative humidity. So friends, each climatic zone or region has different criteria and requirements of stability study, due to which it is very difficult in earlier time to register product in different climatic zones or regions. To overcome this difficulty, the ICH has published its first harmonized stability guideline, that is ICHQ1A Stability Testing of New Drug Substances and Products. This guideline is mainly applicable for Zone 1 and 2. The principle of this guideline was established and harmonized for 3 regions, that is Europe, US and Japan. However, this principle is followed across the world. Now, what is stability procedure? The stability study should include how to select number of batches and batch size, how to select container closure system, what are testing intervals and storage conditions, what type of study or test to perform, and preparation of protocol for stability study. So, the stability study should include following major steps such as selection of number of batches and their batch size, selection of container closure system, charging of samples in stability chamber as per storage conditions mentioned in protocol, sample withdrawal as per intervals mentioned in protocol, sample analysis and evaluation, and establishing shelf life. Generally, the stability studies are carried out through approved protocol. The protocol should contain the reason for stability study in brief. It should also contain the details of API source, its vendor details, batch number, size of batch, etc. Then it should mention the details of container closure systems such as primary packaging material used for packaging of drug substance or drug product. Then it should define the storage condition, testing intervals, also type of test to be performed, test procedure and their acceptance criteria. Now we will discuss steps of stability study one by one. So first is selection of batches. In this we will understand what is the criteria to select batches for stability study of drug substances and products. As per ICHQ1A R2 guideline, in both the cases, that is drug substances and drug products, the stability studies data on minimum 3 primary batches shall be submitted for registration purpose. For drug substance, the batches should be manufactured to at least pilot scale, that is 3 primary batches should be pilot scale batches. The batches should be manufactured by the same synthetic route as production batches. Similarly, in drug product also, the stability studies data should be provided on at least 3 primary batches. Out of 3 primary batches, 2 batches should be manufactured to at least pilot scale and 1 small scale batch can be acceptable. The primary batches should have same formulation and packaged in the same container closure system to be used for marketing purpose. The manufacturing process used for manufacturing of primary batches should be similar to the manufacturing process for intended commercial batches. The overall quality of the primary batches should be equivalent to the intended commercial batches. Primary batches of the drug product should be manufactured by using different lots or batches of the drug substance. Minimum 2 different lots of drug substances are required for manufacturing of 3 primary batches. The stability study should be conducted for each strength, each type of container and pack size unless it is not covered under bracketing or matrixing. The bracketing or matrixing are reduced designs for stability study, that is, matrixing or bracketing where the testing frequency is reduced or certain factor combinations are not tested at all can be applied if justified. Now, what should be the batch size of pilot scale batch? Minimum 10% of intended commercial batch size or 1 lakh unit, whichever is greater, is considered as pilot scale batch. And the batch size of smaller batch should not be less than 25% of the pilot scale batch. For example, in case of A, if your batch size of commercial batch is 10 lakh tablets, then 10% of 10 lakh would be 1 lakh tablets, which is equal to the criteria of 1 lakh unit. Hence, this can be considered as a pilot scale batch size. Or in case of B, if your batch size of commercial batch is 15 lakh tablets, then 10% of 15 lakh would be 1.5 lakh tablets. In this case, 1.5 lakh is greater than 1 lakh unit tablets. Hence, in this case, 1.5 lakh can be considered as pilot scale batch size. Next is container closure system. Should be the container proposed for storage or distribution, including any functional secondary packaging. For drug substances, the stability studies should be conducted on drug substance packaged in a container closure system proposed for storage and distribution. For drug product, stability testing should be conducted on the dosage form packaged in a container closure system proposed for marketing, including any secondary packaging and container label. Each strength and container type, pack size should be studied unless bracketing or matrixing is applied. The storage orientation of the product, that is upright versus inverted, may need to be included in a protocol when contact of the product with the container closure system may be expected to affect the stability of the product's content. Generally, this consideration is only for liquids and semi-solids. Now our next question is, what is testing frequency? For drug substance and product, the frequency of testing should be sufficient to establish the stability profile of the drug substance and drug product. There are three different types of stability studies: long-term stability study, intermediate stability study, and accelerated stability studies. The long-term stability study shall cover the entire retest period or shelf life under the recommended storage condition. Intermediate testing stability study is designed to moderately increase the rate of chemical degradation or physical changes of drug substances or drug product. The accelerated stability study is designed to increase the rate of chemical degradation or physical change of drug substance or drug product by using exaggerated or elevated storage conditions. The testing frequency for long-term stability study should be normally every 3 months for the first year, that is 0 month, 3 months, 6 months, 9 months and 12 months. Then every 6 months for the second year, that is 18 months and 24 months. And then every year till the proposed shelf life, that is 36 months, 48 months and 60 months. Which means for first year, you have to analyze your sample quarterly, then for second year 6 monthly and then annually till the proposed shelf life of that product. Next is intermediate stability study. The intermediate stability study is 12-month stability study. Minimum of 4 time points including the initial and final time points, that is 0 month, 6 month, 9 month and 12 month study is recommended. Last is the accelerated stability study. It is 6 month stability study. A minimum 3 time points including the initial and final time point, that is 0 month, 3 month and 6 month study is recommended. Now we will check what are the storage conditions in general case. Different storage conditions are recommended for stability studies based on different cases. In general case, long-term stability studies are conducted at 25 degrees centigrade plus minus 2 degrees centigrade and 60% RH plus minus 5% RH, or 30 degrees centigrade plus minus 2 degrees centigrade and 65% RH plus minus 5% RH. Intermediate stability studies are conducted at 30 degrees centigrade plus minus 2 degrees centigrade and 65% RH plus minus 5% RH. And accelerated stability studies are conducted at 40 degrees centigrade plus minus 2 degrees centigrade and 75% RH plus minus 5% RH. In certain cases, particularly in case of zone 4, that is hot and humid or hot and very humid, the long-term stability studies are generally conducted at 30 degrees centigrade plus minus 2 degrees centigrade and 65% RH plus minus 5% RH, or 30 degrees centigrade plus minus 2 degrees centigrade and 75% RH plus minus 5% RH condition. In this case, there is no intermediate condition. Hence, the asterisk mark is given for it is up to the applicant to decide whether the long-term stability studies are performed at 25 degrees centigrade plus minus 2 degrees centigrade and 60% RH plus minus 5% RH, or 30 degrees centigrade plus minus 2 degrees centigrade and 65% RH plus minus 5% RH. and the double asterisk mark is given for if 30 degree plus minus 2 degree centigrade and 65% RH plus minus 5% RH is the long-term condition, then there is no intermediate condition. Now, considering the length of this video, we will stop here and continue our discussion in next video, where we will discuss different cases of storage conditions and testing frequency for stability study, then significant change, stability commitment and specification. Hope this is useful. Thank you for watching and see you in the next video.